Great. Thank you very much. Thank you for the very kind introduction, Trevor. Very excited to be with you guys today. So I'm going to talk about cell-free DNA and some of our recent AI work for liquid biopsy detection for early cancer and prediction of cancer treatment response. So here are my disclosures. So cell-free DNA has been a game-changing bioanalyte. It's already revolutionized one branch of medicine. It's revolutionized OB-GYN to the point that non-invasive prenatal testing is well within the standard of care. It's currently revolutionizing oncology. We just had our, the field had the first FDA approval of a cell-free DNA test for colon cancer screening just this last week, a test from Guardant. So congratulations to Guardant, and huge congrats to our field as we're moving further and faster in liquid biopsy cell-free DNA revolutionizing oncology. So, this is a slide that, seemed very futuristic when it was published back in 2017 by Nitzan Rosenfeld's group, but now we can really feel it. We can feel it kind of cell-free DNA touching every single aspect of this timeline, all the way from early cancer screening, MRD detection, monitoring, and clonal evolution. So I'm going to start by talking about one of our early detection projects. So this is a really cool project where we're focusing on NF1 malignant peripheral nerve sheath tumors. Can we detect a cancer in patients with a cancer predisposition syndrome? Can we begin to detect the malignancy compared to the pre-malignant entity? And NF1 MPNST, 1 in 3,000 live births have NF1. About a third of them develop benign plexiform neurofibromas. About half of these transition to MPNSTs. And the kicker is that when patients develop this, and unfortunately too many of these patients do, it's a highly morbid disease that causes a lot of pain because it sits on the nerve sheaths with an 80% 5-year mortality. And then the other thing is that these are kids, people who have a diagnosis of NF1 from the time of birth. So they are getting exquisite follow-up by a pediatrician, followed by an adult family care physician with a strong genomics and genetics background. But nonetheless, so many of these, around 10%, develop MPNST within their life. So clearly we're not doing a good enough job in identifying these and anticipating them and then forestalling what's often a deadly outcome. So why is that? Well, a couple reasons. One is that on physical exam, the pre-malignant and the malignant entities look similar. On MRI, they look similar. Under the microscope, they do look different. So you see increased mitotic figures in the patients with MPNST compared to a more benign-looking histology in the pre-malignant entities. And then genomically, we see a lot of aneuploidy and copy number alterations in the patients with MPNST compared to the plexiform neurofibroma and atypical neurofibroma. So imaging is nonspecific. Tissue biopsies can be complicated by intratumoral heterogeneity. And so, we set up this collaboration with my previous group at WashU, my current group at Mayo, Angela Herby at WashU, and Jack Shern at the NCI. And we put together a study where we compared cell-free DNA from healthy volunteers to NF1 with plexiform neurofibroma with NF1 with malignant peripheral nerve sheath tumor, focusing on copy number alterations, focusing primarily on genome-wide copy number alterations. And I'm showing here that in the tumor tissue DNA, we saw genome-wide copy number alterations which were reflected in pre-treatment cell-free DNA and were more or less blunted in post-treatment cell-free DNA after an effective treatment, and then the genomic DNA profile is rather flat, as you would expect. Cell-free DNA copy number alterations were in canonical regions of the genome as well. We're seeing 8q, 9p, SUS12, CDKN2A, regions that we would expect in NF1 MPNST to be altered. That's what we're seeing. And this is data that we analyzed using the iCourse CNA platform from Victor Aadel Steenson's group from the Broad Institute. So again, same way to just look at the same data I showed before. In a heat map form, we're seeing canonical regions being altered in MPNST, much less so in plexiform neurofibroma or healthy. But then the issue is just looking at copy number alterations, we weren't able to distinguish MPNST from plexiform neurofibroma with high accuracy. The area under the curve was only about 0.6, so it's kind of failing in terms of being a robust classifier. So we wondered we wondered why that is, and is there some other feature of cell-free DNA that we can use to improve our classifier? And we reminded ourselves that it's been shown by multiple groups that cell-free DNA from cancer patients is shorter than cell-free DNA from healthy donors. This is, I'm showing here some figures, the one on the left from Jay Chaudhuri's group, the one on the right from Nitzan Rosenfeld's group, same story. ctDNA coming from cancer is shorter than healthy cell-free DNA. And so we looked in our data, and strikingly, MPNST patients had shorter cfDNA fragments than plexiform neurofibroma. So we're extending that previous finding of cancer versus healthy to cancer versus precancer. We're seeing shortening of the cell-free DNA fragments in the MPNST patients compared to the patients with just the plexiform neurofibroma precursor. And here is really nice data from Trevor's group, which in kind of an even more canonical cancer predisposition syndrome of Lee-Fraumeni syndrome, they're seeing shortening of the active cancer patients compared to the LFS healthies, and then healthy donors without LFS have longer cell-free DNA fragments. A really nice stratification just based on cell-free DNA fragment size alone. Anyways, so when we focused on copy number alterations within shorter fragment sizes, less than 150 base pairs, then we're able to stratify, and here I'm showing the difference. Without size selection, AUC of only 0.6. With size selection, the AUC goes all the way up above 0.8 with accuracy and 85% plus, and nicely comparable to the most advanced imaging modalities that are available today. So this was really the insight, kind of the cell-free DNA genomic insight that saved this project, this idea of size, shorter size of MPNST versus plexiform neurofibroma. More recently, we've extended this to also look at atypical neurofibroma, which is an intermediate form in between plexiform and MPNST. We're seeing an intermediate size of atypicals between PN and MPNST. And we also asked, are there more fine-grained cell-free DNA fragmentomic differences between NF1 MPNST and plexiform neurofibroma and atypical neurofibroma? So to do this, we put together a study of 167 patients, so really the largest liquid biopsy cell-free DNA study in NF1 ever, in collaboration again with WashU and with the NCI, spanning healthy volunteers, plexiform neurofibromas, atypicals, and MPNSTs. And we started by looking at bin-wise fragmentomics. Can we walk across the genome in 5 megabase bins and essentially look at the short to long fragment ratios? And when we do that, we see much greater aberrancy, kind of fragmentomic aberrancy in MPNST compared to healthies, compared to plexiform neurofibromas, and compared to atypical neurofibromas, where the switching from, it's not just yes, there's overall shortening, but there's a lot of switching in terms of short to long as you walk across the genome. This concept of fragmentomic aberrancy is really quite apparent in MPNST compared to the other entities. So when we use bin-wise fragmentomics, and here we're adapting the Delphi approach, which was developed by Victor Velculescu's group and Robert Scharf's group over at Johns Hopkins. So here we're applying the Delphi approach, this bin-wise fragmentomics approach, to our data, and we're able to develop a better accuracy of the test in terms of MPNST versus plexiform, MPNST versus healthy, than we were just using our size-selected copy number approach from our earlier paper. And I'm showing here an example on the left of a patient who had rapid growth of a tumor that was resected, clinically suspicious for malignant peripheral nerve sheath tumor, for MPNST, given the clinical picture. However, when resected, was purely an atypical neurofibroma, and we're able to predict that using this bin-wise fragmentomics approach. We've also looked into NMF, non-negative matrix factorization. Here I'm just, many of you may already know this, but I'm just showing this, just for your edification. with NMF, we're essentially looking at a signature of healthies versus tumor. We know that the tumor signature from our prior work and from others is shorter, the healthy signature is longer. So can we essentially go through our data, our fragmentomics data, and assign weights in terms of how much of this longer healthy signature versus how much of this shorter tumor cell-free DNA signature are we seeing with non-negative matrix factorization deconvolution? So applying this NMF approach, looking at, deconvolving these two states, this healthy state versus this MPNST state, we're able to achieve, pretty whopping area under the curve across the different entities, MPNST versus plexiform, MPNST versus atypical, even atypical versus plexiform, and even healthy versus plexiform neurofibroma. So you can imagine, with this approach, we're starting to see evidence that we can start to track a patient going all the way from the first inception of the development of the plexiform neurofibroma to the development of the pre-malignant atypical neurofibroma, going onwards to MPNST. The NMF score is also correlated with our original ICOR tumor fraction, so our original size-selected tumor fraction from our earlier paper, showing that there's likely underlying similarity in the biology. Of course, we're seeing fewer false negatives with this NMF deconvolution approach. And here I'm showing an example. This is a patient who had, had a tumor that was biopsied for atypical neurofibroma. However, when it was resected with a narrow margin resection, right next to the atypical neurofibroma, there was an MPNST that was simply not, the core needle biopsy needle didn't pierce the MPNST, so it was an inaccurate diagnosis histologically, and as a result, the patient underwent a narrow margin resection with a positive margin. With both the NMF approach and our original size-selected copy number approach, we detect the persistence of MRD, and that's consistent with the patient developing a relapse. So had we known this information using our liquid biopsy approaches, that the patient actually seems to have MPNST up front rather than atypical, then maybe the clinicians would have offered a wide margin resection, and this could have prevented the recurrence, theoretically speaking. Now, CFDNA fragmentomics does encompass more than size. This is a nice figure from Dennis Lowe's paper, from a review article that he wrote in 2022. And in that vein, we've also started looking at end motifs, and we've looked at them in different waves. We've looked at the motif signatures themselves, the 4 nucleotides at the ends of each cell-free DNA fragment. We've also looked at the motif diversity score. Can we look at Shannon entropy of these former end motifs? We've also done deconvolution, NMF deconvolution of these end motifs, and looked at what Dennis Lowe refers to as founder profiles in his paper on the topic. So applying these different methods, I'm showing proof of concept data here that we can start to distinguish plexiform neurofibroma from atypical neurofibroma using these end motifs. Now here we're showing across these different modalities our original ICOR CNA size-selected tumor fraction approach, bin-wise fragmentomics, end motifs, and NMF deconvolution. And you're seeing here the power of cell-free DNA fragmentomics for distinguishing NF1 disease states from one another. And we envision this, being translated in the future, hopefully, our group and collaborators such as Trevor's group, can we really take this forward, to a future standard of care, which is far more practical. Because right now, we know that clinical exam is not good enough. Imaging is, not good enough either, and imaging in kids is very challenging. Can we use a simpler blood test in order to identify these patients who are at risk for pre-malignancy and malignancy? So I'm now going to pivot. I'm going to pivot pretty hard, and I'm going to talk about something later on in this kind of spectrum, going from early detection forward to metastatic disease. I'm going to talk about some of our work in the immunotherapy response space that I'm super excited about. So we're all familiar with exome-wide tumor mutational burden. It's a biomarker for immunotherapy response. It's been shown by multiple groups. Here I'm showing the kind of the landmark paper from Tim Chan's group in the New England Journal of Medicine showing TMB as an immunotherapy response biomarker. So here, this is work that I completed during my residency, kind of applying this TMB approach to a targeted hybrid capture panel. At this point, I think this is quite common in the commercial space, but this was kind of the first example of showing that we can use a targeted hybrid capture NGS assay in order to interpolate TMB. A year later, there was a nice paper from Foundation Medicine showing the same results, that you can interpolate blood-based TMB from a hybrid capture panel, and that correlates strongly with exome-wide TMB. Patients who have elevated blood TMB do better with immune checkpoint blockade, and this was really nicely validated in the prospective BFAST study more recently, published by Peters and colleagues in Nature Medicine. Some other work that we've done is we've also tried to translate this sort of technology into urine. So this is a project where we're looking at patients with bladder cancer. We extract urine cell-free DNA, and then we measure TMB from the hybrid capture panel using the same sort of approach that we published earlier. And we're able to infer TMB from the urine tumor DNA analysis, including patients who have elevated urine-based TMB, including identifying patients who seem to be more likely to benefit from adjuvant immune checkpoint blockade, such as the patient on the right who had elevated urine TMB, received adjuvant immune checkpoint blockade, and remained disease-free. We've also worked with the Friends of Cancer Research, focusing on whether changes in ctDNA reflect response to immune checkpoint blockade. And here in this study of 200 patients across 5 different studies and 5 different independent technologies, we saw overall that when you have an increase in ctDNA going from pre-treatment to on-treatment, those patients have worse PFS, while when you see a decrease, they have improved PFS, and intermediate is intermediate, really nicely corroborating Trevor Pugh's earlier work published in Nature Cancer showing the same pattern. So more recently, we've been asking, can we track a feature of cell-free DNA that is more mechanistically tied to immunotherapy cellular response in order to predict immune checkpoint response pre-treatment? And so we wondered, what could this be? I mean, ctDNA is very exciting, but ctDNA, in order to measure a difference in ctDNA, you need to measure the difference, meaning that you have to measure from pre-treatment to on-treatment. But wouldn't it be nice if upfront, pre-treatment, we could identify these patients who are going to respond to treatment versus the patients who simply are not going to respond to treatment, and we can prioritize them for something else. So to do this, I worked with my good friend and colleague Aaron Newman at Stanford, and we co-developed this approach, which we're calling liquid TME, which starts with some of Aaron's really deep expertise in tumor deconvolution and spatial transcriptomics, where he started by performing spatial transcriptomics across several different carcinomas as well as melanoma in order to identify essentially, what seems like a Google Maps of different spatial transcriptomic ecotypes, going from farther out from distance from the tumor cells in the tumor microenvironment to closer to tumor cells within the tumor microenvironment. And we correlated that with immunotherapy response. Which of these spatial ecotypes correlate more with immunotherapy response, and which correlate more with immunotherapy resistance? And then working closely with my group, we tried to translate that technology to the cell-free DNA space, and I'm going to show you some of that work with the goal of stratifying immunotherapy response outcomes. So to do this, we developed a neural network approach in order to quantify these spatial ecotypes in plasma methylated cell-free DNA. And I'm showing here data from paired tumor samples and plasma samples with EM-seq, and we're quantifying these spatial ecotypes between them. Overall, we're seeing nice correlations. I'm going to zero you in on these two spatial ecotypes of a special interest, SE10 and SE4. I'm going to show you why they're so interesting. So looking across both tumor tissue data and cell-free DNA data, we see a nice correlation in terms of immunotherapy response versus resistance across these different spatial ecotypes, where SE10 is most associated with immunotherapy response and SE4 is most associated with immunotherapy resistance. And I'm showing here across 80 melanoma patients that we banked in-house between WashU and our collaborators at Yale. At the top, you're seeing immunotherapy response versus resistance. NDB stands for no durable benefit. Those are our progressors. DCB stands for durable clinical benefit. Those are our responders. We see ctDNA, which we profiled using the very nice Roche Avenio platform, which I'm a very big fan of. But unfortunately, we see that, as you would expect, baseline ctDNA levels are not stratifying DCB versus NDB. However, when we look at cell-free DNA spatial ecotypes of SE10 versus SE4, you see that SE10 correlates quite nicely with immunotherapy response, and SE4, interestingly, correlates with immunotherapy resistance. So here I'm showing the data another way. You're seeing NDB versus DCB, so again, the progressors versus the responders. The progressors are in blue, the responders are in orange. And you see here with SE10, you see a boost in the responders compared to the non-responders, whether we're looking at all samples or stratifying with just anti-CTLA-4 and anti-PD-1 combination therapy or just anti-PD-1 single checkpoint blockade. And overall, when we stratify by SE10 levels, the patients who are SE10 high in their blood plasma at baseline have improved progression-free survival compared to the ones who are SE10 low. We see the flip when we look at SE4. So SE4, as you may recall, correlated with immunotherapy resistance. So in this case, we're seeing an elevation of SE4 in plasma cell-free DNA in the non-responders compared to the responders. And as you associate with that, looking at PFS, the patients who are SE4 high in their blood plasma have worse PFS than the ones who are SE4 low. So we've also been very interested in immunotherapy toxicity prediction, again, working very closely with Aaron Newman at Stanford, also working closely with Ruth Halaban at Yale. We published this paper in Nature Medicine in 2022. We're actively working on the subsequent version of this technology and looking more deeply at the underlying biology. But long story short, what we identified was that there is an enrichment of activated CD4 memory T cells as well as T cell receptor diversity in the peripheral blood at baseline of patients, of melanoma patients who ended up developing severe immune checkpoint blockade toxicity, what we call immune-related adverse events or IRAEs. Severe meaning toxicity that lands them in the hospital or the immune checkpoint or the ICU or causes death. These patients had significant elevation of either activated CD4 memory T cells that tended to be clonally diverse. Combining these two features of activated CD4 memory T cell abundance and T cell receptor diversity using logistic regression, we come up with what we are calling the composite model, and patients with an elevated composite model score are more likely to develop toxicity, and patients with a lower composite model score at baseline are less likely to develop severe toxicity, as shown here. Interestingly, we see a dose-dependent effect where the higher the composite score, the higher their grade of toxicity, going from asymptomatic to symptomatic to severe to life-threatening, and also the higher the burden of toxicity. So as the scores get higher, the patients have more organ systems that are involved with toxicity. And interestingly, the composite model score also predicted the time to severe toxicity, where patients with an elevated composite model score had a shorter time to severe toxicity than the ones who had a low composite model score. So in summary, liquid biopsy coupled with AI has enormous potential to predict early cancer detection and immunotherapy response and toxicity. Circulating tumor DNA can be used to detect hereditarily predisposed cancer patients in the setting of NF1 peripheral nerve sheath tumors. Fragmentomics, as we've shown, as Trevor's shown in his group, can discriminate plexiform neurofibroma disease states, including pre-malignant atypical neurofibromas. Plasma cell-free DNA methylation features plus neural networks can facilitate immunotherapy response prediction in melanoma patients. And blood-based activated CD4 T effector memory cells and T cell receptor diversity can be used to predict immunotherapy toxicity. So with that, I'd like to acknowledge members of my lab group. This was at my student Noah's MSCP thesis defense, which was in April. Members of my lab who contributed, their names are in blue. My funding sources, my collaborators who I acknowledge along the way during the talk, who are absolutely instrumental in this work, and most importantly, patients, their families. Thank you very much.
Yeah, fabulous talk, Aadel.
Thank you.
I want to jump in with the first question. There's so much clinically relevant biology you were able to pull out of the bloodstream. What's the barrier to move from all these findings to a clinical test? I just think especially for this audience, where that's kind of our job, how do we make that leap?
I mean, it's a fantastic question, Trevor. I mean, I think that I think there are a couple things. So one is kind of, can we expand out to larger cohorts to kind of to prove multi-institutional validation of some of this very interesting work. Another thing is, can we kind of work with the different, regulatory bodies and, make sure that we're developing clear versions of these assays and moving stepwise through the process, the process that you guys understand much better than I do as more of a researcher. I think that the phase of the field where we are still trying to convince ourselves and convince the regulatory bodies, convince the scientists, convince the clinicians that cell-free DNA matters. I think we are now beyond that phase. I mean, if I was giving this talk 3 years ago, I would say that's also important, that we need to also convince our clinical and scientific colleagues. But I think now the work from your group, the work from mine, the work from all of you in the audience, that we've done that convincing. So now it's really going stepwise, clear versions of the assays, working with the regulatory bodies and getting FDA approval and getting this out to the clinic.
Are there any online questions? I don't have eyes on those. I did have another question. Do you think the entire genome is in the blood to be found? Like, if we just sequence the life out of it, could you get an entire karyotype or copy number track?
I think so. I mean, I think that, we know that there are peaks and valleys which relate to nucleosome positioning, so there are going to be some regions that are going to be very shallow, going to have very shallow depth, because those are regions of the genome that are being transcribed. They're open chromatin, and they're going to be less represented in cell-free DNA. But I think if you sequence deeply, I think we can fully karyotype from plasma cell-free DNA.
Yeah, look forward to a future talk on that topic. Great. Oh, we have a question at the microphone. We've only got 30 seconds. So.
Yes,
sorry about that.
I just want to congratulate on your wonderful work. I think the utility of circulating DNA in breast, ovarian, prostatic cancer, even colorectal cancers has come out very well, and we are using it routinely in clinical practice. But the involvement in CNS tumors was something which we were waiting for that, and since I've seen it for the first time, and it's congratulating that you're able to calculate the TMB values for the options for immunotherapy. So my question to you is, are we ready for clinical use now? Can we practice it?
We're not ready quite yet, and this is these are still kind of do not post slides from my research lab, but we're in the process of I think we're at the earliest stages of that that timeline. But our goal is to do everything we can to hopefully get this into the clinic.
Thank you, and congratulations to you.
Thank you very much.
Join me in thanking Dr. Chaudhuri. I believe we're on a lunch break now. Do I have that right, Jane? Oh, roundtables, right. See you there. I'm chairing one. We'll run down to the next room.